To begin our unit on genetics, I want to introduce you to some of the ways that genetics can be used in your practical lives and in the not so distant future. And I think this can help generate some interest in this topic. So in this video in particular, we're going to talk about genetic engineering. The goal is for me to describe the process of gene editing and also cloning. And then we're going to analyze the ways that those processes can be applied to real life and some of the ethical concerns that surround each of those processes. So first, what is genetic engineering? Genetic engineering is the deliberate modification of genetic material, specifically the DNA. And the DNA gets manipulated or changed through different tools and techniques. The two major techniques that I'm going to focus on are going to be CRISPR. That's the first technique. CRISPR, that can be used to edit or change genes, change some of the letters in those genes, the A's, T's, C's, and G's. And the second technique is what we call nuclear transplantation. And that's what gets used to clone organisms, to make clones. Okay, so let's talk about that first technique first, CRISPR. How does it work? So think of CRISPR like a molecular pair of scissors that can find a specific place in the DNA, a specific sequence of letters in the DNA, and it can make cuts and then change the DNA. So we can think of this process as proceeding in 4 basic steps. The first, step 1, the scientists create what's called a guide RNA that matches the piece of DNA they want to modify. Then, step 2, that guide RNA gets added to a cell along with a protein called Cas9. Cas9 is what will cut the DNA. Next, in step 3, the guide RNA finds the target DNA sequence, and that Cas9 cuts that target DNA sequence out. Step 4, the last step, another piece of DNA is swapped into the place of the old DNA, and the enzymes repair the cuts. And so you've essentially taken a piece of DNA out that you don't want, and you put a new piece of DNA in that you do want. That's the big picture of this process. Now, what might CRISPR be used for? What practical applications are there? Well, it can be used first to cure genetic diseases like cystic fibrosis or sickle cell anemia or Tay-Sachs disease. All of these have a genetic basis. They're all caused by certain letters in the DNA. If you can just take those letters out and put different ones in, you could potentially cure these diseases. A second application is that CRISPR can be used to treat cancer. Many cancers are caused or driven by mutations in genes that are called oncogenes. And oncogenes promote uncontrolled cell growth. The cells just keep rapidly dividing. And this is what leads to cancer. If CRISPR was used, you could potentially take out those oncogenes, which would cause cancer. Also, you could use CRISPR to improve agriculture. Their crops, lots of fruits and vegetables and things, these have DNA in them, and the genes in the crops can be edited. You can change them to be resistant to pests or diseases or even harsh environmental conditions. You could change the genes to where the plant can still grow even in an area, say, that's not getting a whole lot of rain. You can also, a final application here with CRISPR is you can enhance traits in livestock. So you could edit the genes in cows to increase their milk production. And so you're producing more milk. Or you could edit the genes in chickens to increase breast size, and then you would have more meat, and so on. So these are some very practical sorts of applications that you could use CRISPR for. And I intentionally, on this slide, I put all the things that look like they could be very good sorts of applications. But there are some concerns about using CRISPR to edit genes. These are the ethical sorts of concerns. The first would be, what about the unintended consequences? So there could be unknown harmful effects on other parts of the organism's DNA. So if you change a part of the DNA here, it might have a negative impact on the organism's DNA somewhere else. And also, this could happen to the DNA in the sperm or egg cells. The reason that's of particular concern, that could harm one's offspring. So it's not just a change to your genes and you're only worried about what it could do to you. It could potentially change the genes that you would pass on to your offspring, and then it could potentially harm somebody else, your children. Okay, a second concern is social inequality. So CRISPR potentially could be used to edit genes in sperm and egg cells, allowing you to design babies to have specific traits like be a certain height or have a certain amount of strength or have a certain level of intelligence. And very likely the only people that would have access to CRISPR, the ability to edit their genes, would be the rich, because this is an expensive process. And think about the kind of inequality that results from that. The rich people are the ones that get to design babies to be stronger, taller, maybe more beautiful, more intelligent. And the poor people who don't have access to gene editing are going to be not in as good a position, not as likely to be as tall or as strong or as beautiful or as intelligent, and so on. So this could lead to social inequality. And basically it leads to a system where the rich most likely just get richer and the poor get poorer. Okay, so those are some ethical concerns about gene editing. What's interesting now is that there's already some of this going on even today. So in certain countries, you can select the biological sex of your baby, whether it's going to be a boy or girl. Now you can do that now through a company called Microsort. And what happens is Microsort separates out sperm from eggs, and then they just ask you, do you want to have a boy or a girl? If you say boy, then they'll use a sperm that has a Y chromosome in it, which carries the genes for making boy parts. And if you say you want a girl, then they'll give a sperm that has an X chromosome in it, which has the genes for making girl parts. Okay, all that was that first technique for editing genes. I said there was a second technique, and that was nuclear transplantation, and this is what gets used to clone organisms. So first, what is a clone? A clone is an individual organism that's genetically identical to another organism. And clones can be and have been made through a process called nuclear transplantation. I put an image here of Time magazine from many years ago now, where they cloned a sheep named Dolly. And essentially what happens, I'll go through the process here. Oh, let me back up slightly and explain this image first. So what happens is you have two sheep, a female and another female, and you take one of the sheep's body cells. So let's call this sheep A. You take sheep A's body cell. So that could be a muscle cell, that could be a skin cell, one of the body cells from sheep A, and you extract the DNA from it. Okay, so that gets us to having the DNA of sheep A. Now, you take sheep B, you take an egg cell from sheep B. It has to be an egg cell. And you remove the nucleus. So now, once you remove the nucleus, that the DNA that was in the egg cell isn't there anymore. And all you do is you put into that cell the DNA from sheep A. Once you do that, that cell is going to be used to create this sheep C. So what you do, or I'm sorry, that cell is going to be implanted into the uterus of sheep C. And once it does that, when that sheep gives birth, when sheep C gives birth, the baby that's born, the lamb, will just be a clone of sheep A. So that's how they were able to make a clone of Dolly. Now, that process is what you call reproductive cloning. But there's another technique for cloning called therapeutic cloning, and I'm going to show you the differences here. So in therapeutic cloning, the cloned embryo is not implanted into the woman's uterus for pregnancy. Instead, the embryo is allowed to develop in a laboratory dish, and from that you just harvest stem cells. And those harvested stem cells, those are what get placed into specific environments or are given specific signals that cause the cell to differentiate into specialized types of cells. So you can see up here, you've got a clone. So you've got these cells that rather than you're putting them in a woman's uterus, you actually just leave them in the lab dish. You allow them to develop into specific kinds of cells, and then potentially you can use those to place in someone's body, and then they could potentially have new nerve cells or new blood cells or new muscle cells from this process. That's therapeutic cloning. Reproductive cloning, like I said on the last slide, you actually implant the embryo in the uterus of the mother, and then the baby that's born would be the clone. How can cloning be used in real life? It could be used to save endangered species. You could clone organisms that are at a dangerously low population. Cloning can also be used to treat various medical conditions. For example, the need for organ donors would be decreased if we used cloning, potentially at least. And the chance of a successful transplant gets increased because there would be an exact tissue match, since you use the genes from this person that needs the transplant. You use their genes to create a new organ for them, like a new heart or a new kidney, and that would make an exact match between them. Sometimes what happens when a donor is not identical to the person receiving the organ, sometimes the body of the person that's receiving the organ, their body just rejects it and won't use it. But that happens, that the chance of that happening would go way down if there was an exact tissue match, which you could get through cloning. Also, diabetes and Parkinson's disease could be treated using cloning if we transplanted specialized cells that function properly to replace the damaged cells in the pancreas or in the brain. Now, there are some ethical concerns about cloning. One is the destruction of embryos. So, research on cloning often requires the destruction of embryos, and there is a debate about when human life begins. And if it begins at conception, then does this embryo have a human right to life? A second ethical concern is, what about the rights of clones? Does a clone have their own unique identity and rights, or are they merely a copy of the original person and so require the approval of the original person for various bodily decisions? Another ethical concern, what about unintended consequences? There could be unforeseen genetic or developmental problems in cloned individuals, and if that's the case, should we risk harming them. One more concern or ethical concern is the social inequality. Access to cloning technology will be expensive, resulting in only the rich having the ability to clone. Is that fair? So these are some ethical concerns about cloning.